Hi, we're Sarah, Maya, and Amina, and we did the adrenaline signaling pathway, specifically heart rate. Adrenaline is a signaling molecule in this pathway, and it is hydrophilic, meaning it cannot pass through the lipid bilayer of the cell membrane. Because of this, its receptor protein is located on the cell membrane, not inside the cell. Some advantages of GCPRs include signal amplification and rapid cellular responses. One ligand-bound receptor can activate multiple G proteins, making the response strong even at low signal levels. GPCRs are also very diverse, which allows cells to respond to many different signals and makes them major targets for pharmaceutical drugs. However, there are also disadvantages. GPCRs can undergo rapid desensitization, repeated stimulation can reduce responsiveness, and because they were found throughout the body, activation can lead to unintended side effects. Overactivation may also cause harmful effects, and many GPCR ligands have short half-lives. The receptor involved is a G protein coupled receptor. GPCRs are commonly used in cells because they allow for fast signaling and strong amplification. They work by changing shape upon binding to a ligand, which activates an attached G protein, triggering downstream signaling cascades.
The specific players in the adrenaline signaling pathway include adrenaline, the G protein coupled receptor, specifically the adrenergic receptor, adenylyl cyclase, CAMP, and protein kinase. First, adrenaline is secreted by the adrenal glands as a signaling molecule. Adrenaline then binds to G protein coupled receptors that span across the cell membrane, since adrenaline is polar and cannot cross the hydrophobic part of the membrane. When adrenaline binds, the G protein is activated by the exchange of GDP for GTP. The G protein then breaks into subunits, and only the alpha subunit continues in the signaling pathway. The activated G protein stimulates the enzyme adenylyl cyclase, which converts ATP into CAMP. CAMP acts as a second messenger and activates protein kinase A, or PKA, amplifying the signal. The intended response of the cell is to prepare the body for fight or flight. This includes expanding the lungs for more oxygen, increasing heart rate and blood pressure to deliver oxygen more effectively. This leads to glycogen phosphorylation, causing glycogen to be broken down in the liver and muscles. As a result, glucose levels in the bloodstream increase, providing quick energy. The body becomes more alert, and physical changes may include sweating, trembling, and pupil dilation.
Next, we will explain how the signaling pathway is terminated. The pathway ends when adrenaline dissociates from the receptor. Without the ligand, the receptor can no longer activate. The G protein then hydrolyzes GTP back into GDP and becomes inactive. The inactive G protein dissociates from adenyl cyclase, stopping CAMP production. CAMP is broken down into AMP by CAMP phosphodiesterase, and protein phosphates remove phosphate groups added during activation. This reverses the metabolic changes and returns the cell to its resting state. An example of an antagonist in this pathway is propranolol, which is an exogenous antagonist. Propranolol blocks adrenergic receptors in the heart, muscles, and nervous system, preventing adrenaline and noradrenaline from activating the G protein. This stops CAMP production and prevents the activation of PKA. Because propranolol blocks these receptors non-selectively, it suppresses increases in heart rate and contraction strength, decreasing cardiac work. This is why propranolol is commonly used to treat conditions such as high blood pressure and anxiety.